Nikol, and with my teammates Annika and Ben, we'll be looking at the effect of spaceflight on neuronal development. We've imaged faraway galaxies with groundbreaking technology, but what issues do we still need to address to actually send humans deeper into space? One of the most pressing issues is protecting human health, and our experiments involve neurons, the cells of one of the most important systems in our bodies. A lot of changes happen to the nervous system during spaceflight. This diagram shows spaceflight impacting vision, balance, movement, and sensory processing, all essential to performing tasks and maintaining a good quality of life on long-term missions. To help understand these complex processes, we're investigating specific changes to neurons during spaceflight.
And in a spaceflight environment, we've seen dramatic changes to the human skeleton. But it's not just our bones that are changing. Our cellular skeleton, the cytoskeleton, has also been shown to behave in entirely different ways in space. We'll be looking at one of the most important components of the cytoskeleton, microtubules. The individual strands in this bundle are microtubules made of alpha and beta tubulin heterodimers. They're dynamically unstable, meaning they switch between periods of growth, or polymerization, and shrinkage, or depolymerization. And multiple studies have shown that microtubules are disrupted in microgravity, with shorter and wavier microtubules forming in space compared to on Earth. And this is important because we know microtubules play a really central role in neuronal growth and development. Here, you can see microtubules labeled with green, and they're bundled all throughout the axon and dendrites of this neuron and into the growth cones at the tips of these outgrowths. Not only are they essential structurally, but they also influence growth cone steering. So they're really deeply involved in neuronal development.
And that makes this an intriguing combination of changes. We've seen in space that neurons behave differently, with changes to the quantity and structure of branching. And we've seen in other cells that microtubules behave differently, but this has not yet been observed in neurons. This leads us to hypothesize that changes to the dynamics and structure of microtubules are causing neurons to behave differently in space.
Our ambitious experiment evaluates microtubule dynamics and neuronal development. We'll be using C. elegans as our model organism because the worm's nervous system has been well studied and it is easy to send into space. We'll start our C. elegans at the L1 stage, when they survive freezing best. Send them to the ISS, thaw them, and allow them to develop to the late L4 phase, which is adulthood. We'll do PCR and microscopy on samples from each of the developmental stages. In addition to our microgravity group being sent to the ISS, we'll also have a 1G, or Earth gravity group, as a negative control. And we'll also be treating our C. elegans with the drugs colchicine and paclitaxel in 1G and in microgravity. The drug treatments serve as additional controls, with colchicine, a negative control, used as a tool to cause microtubule shrinkage, and paclitaxel, a positive control, used to cause microtubule growth. We'll do PCR and microscopy on the microgravity and 1G groups, but microscopy only on the drug treatments. For microscopy, we'll start by looking at the general structure of PVD neurons in C. elegans, which we chose because they have an easy to quantify structure. Then we'll increase magnification and look at the structure and position of microtubules within the PVD dendrites. Here you can see a bundle of microtubules with a single microtubule extending out, beginning the turning process that forms the branching above. We'll discuss some of the results we expect from the microscopy section based on existing literature on other cell types. In microgravity, we expect shorter and wavier microtubules compared to 1G, and here's an example of some of the data we might collect on the length of microtubules. In 1G, colchicine causes depolymerization, so we expect shorter microtubules, and paclitaxel causes polymerization, so we expect longer microtubules. In microgravity, we expect that colchicine would increase the depolymerization already caused by microgravity, producing the shortest microtubules, and that paclitaxel would counteract the depolymerizing effect of microgravity, producing microtubules similar in length to the 1G group. In 1G, we expect normal PVD neuron structure, but in microgravity, general disorganization, and of interest to us, an increase in the number of branches. In 1G colchicine, we expect little to no branching, but for paclitaxel, an increase in the number of branches. In microgravity, we expect the same from colchicine, but for paclitaxel, normal PVD neuron structure. In other words, paclitaxel could be used to counteract the depolymerization caused by microgravity and produce normal PVD neuron structure. We also wanted to look at microtubules on a more molecular level with PCR. We'll do reverse transcription PCR on our C. elegans, fluorescently labeling the product so that we can observe it with the P51 fluorescence viewer, indicating the level of gene expression. We'll look at genes associated with microtubules to see if changes in their expression correlate with the changes to microtubules. We'll look at alpha and beta tubulin, but we don't expect a difference in expression between the microgravity and 1G groups. We'll also look at CLS2, the C. elegans equivalent of CLASP, coding for a protein that causes microtubule polymerization. In microgravity, we expect this to be underexpressed because microtubules are depolymerizing. MEI1, or catenin, causes shrinkage, so we expect this to be overexpressed. And UNC116, a component of kinesin, and BICD1, associated with dynin, both code for motor proteins important in PVD neuron branching. We expect these to be underexpressed because branching is disrupted during spaceflight.
And for our second experiment, we'll be looking more closely at why microtubules are acting differently in space, specifically how microtubule self-assembly, or polymerization, might be changing. So recall that microtubules are polymers of alpha and beta tubulin heterodimers. Here, we're using the BioBit system to look at microtubule polymerization. We'll include the coding genes for alpha and beta tubulin, as well as a nucleation seed to help the microtubules start polymerizing. Our coding genes will be translated into tubulin, which will form the heterodimers that arrange themselves into microtubules. And it's great that we can get this to happen, but we still need a way to look at it. And so we'll be using a system called Forster Resonance Energy Transfer, or FRET, to actually quantify polymerization. We'll tag alpha tubulin with GFP and beta tubulin with RFP. With FRET, when alpha tubulin gets close enough to beta tubulin, GFP transfers energy to RFP, causing it to fluoresce. So in an ideal setting, we'd see fully green fluorescence when there is no polymerization, red fluorescence when there's complete polymerization, and a range of yellows and oranges in between. Getting into our expected results and experimental groups, we'll first look at a control group for each fluorescent protein, one with only RFP tagged beta tubulin, and one with only GFP tagged alpha tubulin. We're expecting a dim red fluorescence from beta tubulin and a bright green fluorescence from alpha tubulin. We'll have 3 flight samples and 3 ground samples, each with 3 tubes. Our first group is the conditions I described previously, and here we're expecting to see less polymerization in microgravity based on existing literature. We'll also have a positive control, including paclitaxel, which again encourages microtubule growth. So here we're expecting to see essentially complete polymerization, slightly less so in microgravity. Lastly, we'll have a negative control, including colchicine, which again encourages microtubule shrinkage. So here we're expecting to see essentially no polymerization. But it's important to note that these tubes still aren't entirely green. That's because with the P51 fluorescence viewer, we'll get a bit of background from RFP, even without FRET having taken place.
So for a quick recap, in our ambitious experiment, we expect to see shorter and wavier microtubules when compared to the 1G group, and at a larger neuronal level, more branching and general disorder of the structure. For our freeze and fly, we expect to see less polymerization in space when compared to the 1G group. So where do we go from here? Once we see what effect changes to microtubules and microgravity have on neuronal development, we have several directions for future experimentation. The first of which is looking at other components of the cytoskeleton, such as actin, which is closely connected to microtubules. Also, since our experiment only focuses on neuronal growth, we may not get the full picture for adult organisms whose cells are also undergoing maintenance. This would be especially important to look at because currently only adults are exposed to microgravity on the ISS. And also, we know from studies such as the one with Mark and Scott Kelly that psychological problems associated with stress and isolation do occur in space, but the connection between cellular and psychological changes is not fully understood. If we could establish a link between psychological health and the underlying mechanics of microtubules and neuron dynamics, we could pinpoint more effective drugs for treating these changes. Our experiment also provides novel uses for BioBits, with the first instance of FRET viewing in conjunction with it, and also will help to establish BioBits as a novel, low-cost solution for studying the cytoskeleton. And finally, as we move into longer duration missions, such as the upcoming Artemis missions, we may see the negative effects of spaceflight exaggerated, and it will become increasingly important to have effective drugs and a complete understanding of all processes involved, so we can mitigate or even prevent them, keeping future spacefaring generations healthy and safe. We'd like to thank everyone on this slide, especially our mentor Alex and our sponsor Artie. Thank you very much.